Okay, I think we'll go ahead and get started. Just double checking that everyone can see my screen. Okay. Yeah, I'd like to welcome everyone to the first ever live event that is the beginning of an educational program featuring a series of talks meant to engage the neuroimaging and brain science community to enhance the translational application of computational neuroimaging techniques in promotion of human brain health. So welcome everyone. Thank you for joining us. And just to begin, just some information about the program itself. This educational program is nested within the Lifespan Working Group of the Enhancing Neuroimaging Genetics Through Meta-Analysis, or ENIGMA, consortium. It hosts expert lectures and training workshops, fostering a community dedicated to improving the translational value and advanced computational analyses of neuroimaging data for brain health. This is an inclusive program that welcomes researchers at all levels of neuroimaging skills and from diverse training backgrounds. The program provides opportunities to expand skills, initiate collaborations, and facilitate networking in a welcoming learning environment. Yeah. And just some information about the core team that has put this effort and initiative together. So that includes Dr. Ruiyang and myself as the directors who have organized and put together these series of events that we will be holding, two of which you have already seen some information about, and the first of which will be taking place today. In addition to the two of us, and my name is Shalaila Haas, and I am an instructor at the Icahn School of Medicine at Mount Sinai. And in a little bit before Dr. Guy's talk, I will introduce him further. But the core advisory committee that has also helped us to put all of this together include Dr. Sophia Frangu, who is professor at the University of British Columbia and the Icahn School of Medicine at Mount Sinai, Dr. David Lan at Harvard Medical School, Dr. Daniel Kaiser, research group leader at the Ludwig Maximilian University in Munich, and Dr. Chao Ganyan, professor at the Institute of Psychology at the Chinese Academy of Sciences. So all of these individuals have worked hard to put all of this together for all of you and the neuroscience community, and we look forward to getting feedback from you, learning more, and sharing our passion for brain science. Here we have the first two events that we have scheduled. Again, the first of which is already going to be happening today. So that's the CentileBrain models and introduction. And this will be there will be a talk given by Dr. Ruiyang, after which then Yuetong will be giving a demonstration on how to actually use this platform and provide some examples of the outputs. Then in January, tentatively, sorry, it should actually say January 17. I will correct that, but it's January 17 at the same time as today. We will have the second session that I will lead, where we will talk about utilizing the CentileBrain models in clinical research, providing a case example in individuals that are at clinical high risk for psychosis. Yeah, it is great to see so much interest from around the world for this initiative. This just gives you a glimpse of some of the individuals that have perused the website so far. And of course, none of this would be possible without your interest. So we're very excited to see where this journey takes us. And if you want to hear more about upcoming events, future events, get involved, or have any questions or comments or feedback, you can join the mailing list on the website. That's translational-neuro.org. There's also the links to both LinkedIn and Twitter, where you can also get in touch with us. Okay, so without further ado, today's session titled CentileBrain Models, an Introduction will be led again by Dr. Ruiyang, one of the co-directors of this educational program. Dr. Guy is currently a research scientist at the Department of Psychiatry at the University of British Columbia. His current research is dedicated to studying brain disorders with a particular focus on mood disorders using brain imaging techniques. He actively contributes to the development of computational methods for analyzing neuroimaging data with the ultimate goal of enhancing diagnostic accuracy and individualized treatment approaches for brain disorders. Following Dr. Guy's introduction, Yuetong will provide a demonstration of the CentileBrain website and its capabilities. Yuetong is currently a PhD candidate at the University of British Columbia, Faculty of Medicine under the supervision of Dr. Sophia Frangu. We're looking forward to your presentation.
Okay. Thank you, Shalaila, for the kind introduction. Hello, everyone. Thanks for joining us for our very first event of this educational program. The title of my presentation today is Normative Modeling of Brain Morphometry Across the Lifespan using CentileBrain. I have nothing to disclose. So in this presentation, I would like to first give an overview of normative modeling in the current neuroimaging community. Then I will introduce our present CentileBrain models of brain morphometry measures. And finally, I will show evidence of generalizability and examples of utility of these CentileBrain models. Let's first have a brief overview of the normative modeling with neuroimaging data. So in general, normative modeling is a framework for mapping how individuals deviate from a populational reference pattern. Perhaps the most well-known application of normative modeling in medicine is the baby growth charts in pediatrics. So the left figure shows a growth chart of the circumference of baby girls from birth to 2 years old. With such a chart, we can easily position a specific individual with regard to head the conference as compared to a reference normative population. And we can tell if this baby's head is too large or too small or just the perfect size compared to the healthy babies with the same age. Despite many years of neuroimaging research, we still do not have normative reference values for brain measures. It means that when we have the value of a brain measure, like, for example, the volume of the hippocampus, at this moment, we cannot tell whether this value is within or outside the normal range. Our hope is that we could have a chart or reference pattern just like the right figure is showing. In this pattern, we construct a relationship between a brain measure and the chronological age. Once we have this pattern, once we have a new individual, we can position this individual on top of this reference pattern to quantify to what extent this individual deviates from the normative mean. For instance, the new participant number 1 here represented with a green dot is within the normal range. While the second new participant, number 2, represented with a blue dot is outside the normal range of healthy people. Now we have a very first impression of what normative modeling is. The next question would be why? Why do we need normative modeling in neuroimaging research? Let's first recap the conventional case control approach. In this type of studies, we often compare the means of 2 groups. For example, there is a control group and there is a clinical patient group. When we compare their mean values, their average values, it's very often we reach a conclusion that the 2 groups are not different. But does this conclusion reflect the reality? Well, perhaps not. Because if you zoom in each group, and look at individuals within each group, you will find that there are actually a lot of variation that cannot be captured properly by this case control approach. Here I show a concrete example. At a group level, we can often find that the patients with the schizophrenia have a globally thinner cortex relative to the healthy controls. But if you look at two individual patients of schizophrenia, you will find that they have slightly different cortical thickness signatures, and their deviations from the mean of healthy controls are even more different from each other. This example reminds us we need to have a method to look at individuals rather than just the mean differences of the two groups. As we have shown in the previous slide, normative modeling is a good method to look at the individuals on top of healthy populations as a reference. This is the primary reason we really need normative modeling in the era of precision medicine. So in this slide, I would like to show you a very simple form of the normative modeling. We can formulate the normative model as a regression model. So in this equation, the response variable y is a brain measure. This brain measure could be a morphometric measure from the T1 weighted MRI scans, or a functional connectivity measure from the resting state functional MRI data, etc., etc. The predictors and or the covariant X could be a variable like age, sex, or a cognitive measure, or even a clinical measure, whichever you are interested in. So although in this figure and in our CentileBrain project, we will construct a regression model just like this, representing a brain measurement as a function of age, I want to emphasize here that a normative model is not fixed. It's not only fixed in this form. Actually, as illustrated in the bottom figure, it can also be a model depicting age or other non-imaging variables as a function of brain measurement. The conventional procedure to build a normative model of a brain measure as a function of age is a procedure to build a machine learning model. We first assemble the reference cohort. Usually this cohort is composed of healthy individuals. And this cohort is often called model development sample. Then we choose the regression method and estimate the model parameters with data from this model development sample. The next step is to validate this model with independent samples. Once we can confirm the model performance is at a satisfying level, we can apply this pre-trained model to new samples to quantify how far this new individuals deviate from the normative reference. In the last 5 years, there is a growing body of research with normative modeling in the neuroimaging research community because the wide availability and validity of the structural MRI data. Majority of this work focuses on brain morphometry. Here, I would like to mention two normative models of brain morphometry throughout the human lifespan. The first one was based on over 100,000 healthy participants, and it focused on normative models of several global brain measures, like, for example, the total brain volume. The second one was based on over 50,000 participants, and it modeled the cortical thickness and subcortical volume of the brain. While the community has a lot of endeavors on this topic, there are at least two knowledge gaps. Firstly, normative modeling was implemented using a variety of approaches which have never been systematically evaluated. This is a major knowledge gap because having such a knowledge of the empirical performance of different approaches, different algorithms would be very helpful for us to design new studies and evaluate currently available literature. Secondly, the generalizability of the established brain morphological norms to a broader range of ethno-racial groups has never been explored because we acknowledge that majority of our currently available MRI data represent samples from the weird societies and from individuals of European ancestry or those who self-identify as non-Hispanic whites. So we really need to validate our models on a broader range of ethno-racial groups through international collaborations. So to bridge these knowledge gaps, we built our CentileBrain models. Now let's have a look at our CentileBrain models. So firstly, I would like to mention that the current CentileBrain model is delivered by the Enigma Lifespan Working Group. So it is really, really a team effort. For the next one is about the model development sample. So this sample is composed of healthy individuals from over 80 MRI data sets, including data from our Enigma Lifespan Working Group, data from Human Connectome Project, ABCD study, UK Biobank study, and some other public available data sets. The total number of participants is over 37,000 with 53% of females and 47% of males. From the figure on the right side, you can see a roughly equal distribution of individuals across both sexes. The age range is from 3 to 90 years, which covers the vast majority of the human lifespan. Here, I would also like to mention that our CentileBrain models are sex-specific, just in order to accommodate sex differences in brain morphometry, meaning that we developed our CentileBrains for each sex separately. So we developed normative models of cortical thickness, cortical surface area, and subcortical volume. Specifically for the cortical thickness and surface area, we have values from 68 cortical regions, and the subcortical volume are from 14 subcortical structures. We use the free surfer to generate values of this measurement. Just in case some people are not familiar with free surfer output, we do have a figure here to show the output we have used in this work. So free surfer generally can identify the pure surface and the white matter surface of the T1 weighted MRI scans. In the meantime, it parcelates the entire cortex into 68 subregions according to the desiccant cleaning atlas, and segments the subcortical structures into 14 subregions according to the we call it ASEC atlas. So in these two atlases, different colors, you can see in the brain, different colors represent different subregions of the brain. The cortical thickness is measures the distance between the pure surface and the white matter surface. And the surface area is the area of the white matter surface. The volume, the subcortical volume is the volume of a subcortical region. Those are very instinct measurement. So for each of these three measurements, we systematically evaluate the performance of 8 different regression algorithms that have been implemented in the current literature. These approaches include ordinary least squares regression, Bayesian linear regression, gammas, et cetera, after selection of the optimal algorithm with the best accuracy and the highest computation efficiency, we further tested the current available covariance. This covariance pertain to image acquisition and data quality, free surfer software versions, and global measure neuroimaging measures. We have also conducted a series of sensitivity analysis about the sample size and about the distinct 8 bins. This work has recently been accepted for publication by Lancet Digital Health, and we also do have a preprint version. So if you want to read it before it's coming out from the journal, please scan this QR code. It will lead you there. So next one, we evaluated the 8 regression algorithm with the 5-fold cross-validation. So the results the results presented here and hereafter are based on the normative models of females. And the results of males have the same patterns as females. And the conclusions are consistent across all brain regions. So here I use left thalamic volume, cortical thickness, and surface area of the left medial optofrontal cortex as the exemplars. Also here, we only present the mean absolute area, MAE, as a measure of the model accuracy. In our paper, we also have root mean squared area and explained variance. So they come, and again, the complete results can be found in our paper. As for the results, we observed that the LMS, Gaussian process regression, warped Bayesian linear regression, and the multivariate fractional polynomial regression, these 4 have comparable performance in terms of model accuracy. But if you look at the CPU time, the Gaussian process regression is quite computational demanding, and the MFPR, which is multivariate fractional polynomial regression, is the most efficient method. So we recommend the multivariate fractional polynomial regression as the optimal algorithm. In this slide, we present the results of the model optimization on the MFPR algorithm. So we evaluated the effect of different covariant combinations on the model performance, and we found that for all of the 3 morphometric measures, the free surfer version and the vendor MR machine vendor tab have very minor effect on the model performance. The optimal covariance combination is a non-linear polynomials of age plus the linear component of global measures. For the subcortical volume, this global measure is the intracranial volume. For the cortical thickness measure, this global measure is the mean thickness across the entire cortex. And for the surface area measure, this global measure is the total surface area of the entire brain. Here we have the conclusion. The most important parameters for model optimization are the global morphometric measures. Next one, we tested the effect of sample size on model performance. So in this slide, in each subplot, each line represents the average MAE values of each region as a function of sample size. And in the next slide, we averaged the individual lines of the each individual region. So right now, each line represents the average MAE value across all the brain regions, and the shadowed area represented the variance across these regions. Now we can see a clear pattern that generally the model accuracy plateaus around 3,000, which means that a sample size of 3,000 is likely a safe number for stable normative models of these 3 morphometric measures. Okay, So in this slide, we present a result of distinct age beams. We divided the entire sample into 9 different age groups. And in these figures, each curve represents the distribution of model performance, MAE, across all brain regions. This result tells us the models of different age groups are quite robust because they're very similar if you look at their distributions. And this phenomenon supports the applicability of the models developed in the entire study sample to groups with a more restricted age range and at different points in their life trajectories. So we have the conclusion that we demonstrated the robustness of the model accuracy across all age groups. In this slide, we used the Euler number as a measure of the data quality. We demonstrated that the data quality has very minor effect on the model performance by showing that adding the Euler number to the model does not really improve the model performance. Of course, we acknowledge that our model development sample is of high quality as we went over the quality control procedures very carefully. Again, these results demonstrated the robustness of our CentileBrain models in the presence of different data quality. So in the last section, I would like to present the evidence to show the generalizability of our CentileBrain models and examples of utility of these models. Before showing the results, I would like to introduce a metric from the CentileBrain model, the z-score, which is a measure of division of an individual from the normative mean. I bring this z-score up here because you will hear this metric again and again in the following slides. So now we have our CentileBrain normative model for a brain measure. In this example, let's say it's the volume of the left hippocampus. With this model, we can project our observed volume of a new participant to this model. Here, I use the green arrow to show the observed volume of this person. And we can further calculate the distance between the observed volume and the predicted volume of this person. And this distance shows reflect how far this person's left hippocampal volume is from the normative mean of the healthy population. So knowing that we have a z-score, we can show the next results. So in the first instance, we use a longitudinal sample to test the longitudinal consistency of our CentileBrain model. The sample includes a SLIM dataset from China and a QTAP dataset from Australia. And the average interval for the 2 time points, because it's a longitudinal data, so we have 2 time points of MRI scans. The average interval for the 2 time points of this sample is about 2 years, roughly. In this slide, we demonstrated the stability of the regional z-scores derived from our optimized CentileBrain models. So in each spaghetti plot, the green lines, each green line represent the individual z-scores of the exemplar region over 2 years, and you can see that these z-scores of each participant are quite stable over time. So, and here we also present the MAE and RMAE values. So the MAE and RMSE values at the 2 time points are quite stable as well. We show the first results of first scan with the blue curve, and with the pink curve, we show the results for the second scan. Altogether, these results demonstrated the robustness of the CentileBrain models for the longitudinal data. In the second instance, we want to demonstrate the generalizability of the current CentileBrain models on different ethno-racial groups. So we collated a sample with two ethno-racial categories by self-report or by genetic testing. In the self-reported category, the participant identifies themselves as Black, South Asian, East Asian Chinese, and East Asian Japanese. In the genetically determined ancestry category, genetic testing was used to categorize the participant as with different background as African, a mixed American, East Asian, and European descent. So for the results, in this slide we present the results of self-reported ethno-racial groups. You can see from the two figures that the results of the genetically you can see that the MAE values of these groups are highly correlated with and resemble the model developmental sample. In the next slide, we present the results of groups based on genetics. You can see from the two figures that the results of the genetically determined categories are very consistent with the results of self-reported ethno-racial groups. Altogether, these results demonstrate that our CentileBrain models generalize to various ethno-racial groups. So in the third instance, we use the early psychosis sample from the Human Connectome project to demonstrate the relevance of the current CentileBrain models for mental illness. We did the demonstration with two arms. The first arm is to use these scores from the CentileBrain for classification of healthy and patients with psychosis. And the second arm is to use these scores to predict the symptom scores of the patients. Here, the symptom score, we use the positive and negative syndrome scale, PANSS. As Dr. Shalaila has mentioned in the introduction, that in the next event, she will introduce the utility of our CentileBrain model to a larger sample of individuals with high risk for psychosis. So we have two sections for this instance. So the first part is the results of diagnostic classification. In the left figure, we show the receiver operating characteristic curves for prediction using the z-scores from the CentileBrain model or using the original free surfer output. You can see that using the z-score from the CentileBrain model improved the classification performance. And in the right figure, we show the difference between these two ROC curves. And you can see that this difference is statistically significant with permutation test. So we have a conclusion that the regional z-scores from the normative models outperformed the observed free surfer data for diagnostic classification. That's the first arm. The second arm is for symptom prediction in terms of PANSS. We show in this figure that using z-scores from the CentileBrain model slightly improved the prediction accuracy of the PANSS with a smaller MAE value. And the curve, of course, the curve in the figure shows the results from the 1,000 permutation test, and the dotted line here represents the real delta MAE between using free surfer output and using z-scores for prediction of PANSS. So we have the conclusion that the regional z-scores from the normative model, our CentileBrain model, outperformed the observed free surfer data for symptom prediction. And finally, we have the take-home message. First, we present an empirically benchmarked framework for normative modeling of brain morphometry across the lifespan. Second, we demonstrated the generalizability of our optimized CentileBrain model to different ethno-racial groups. And most importantly, we present a dedicated web portal to provide our optimal model parameters. With this web application, users can freely apply our pre-trained CentileBrain model to their own dataset. I would like to thank everyone in the Frango team as well as the entire Enigma Lifespan working group, because this work is not possible without their supports and contributions. Finally, please scan this QR code to take a look at our CentileBrain website. And thank you for your attention and wish everyone a happy holiday. Thank you.
Great. Thank you so much. Yes, that was an excellent talk. I hope many of you have kind of overview of what this model shows and has shown and how robust it's been shown to be in various different samples. Now we'll go ahead and move on to the demonstration by Yuetong Yu. And he will show us a little bit more about what you can actually do on the platform.
Thank you, Shalaila. Hello, everyone. My name is Yuetong, and today I'm very excited to be here to introduce the CentileBrain website. So what is CentileBrain website? CentileBrain website is a comprehensive platform that allows people to explore the latest research about CentileBrain, access CentileBrain open source scripts, as well as to generate your normative deviation scores from your sample, and so much more. And today, I would like to introduce some important features on our website, including centile curve and MFP curve visualization, online calculation tool, and the script to generate your own CentileBrain models from your own data. So let's just get started. First, I would like to introduce the centile curve and the MFP curve visualization. So when we load the homepage, we see on the top bar, there are different options. When we click the Explore CentileBrain, we can see two figures here showing some demographic information about the CentileBrain training data. And when we switch to the visualization, visualize regional age-related curves, we can explore the centile curves and the MFP curves for different morphometric features across different brain regions in female and female. So here, I would like to take a simple example. So if, for example, if we choose the female on subcortical volumes for the hippocampus, we can see the centile curves here. So for centile curves, it is an interactive curve. So users can hover their mouse on this plot to compare values for different centiles. And you can also visualize all of the centiles at a time. And on the top, on the right part, there's a switch where you can switch from centile curves to the MFP regression curves. So here, users can explore the trends and all training data points we used for the MFP model. So the second feature I would like to introduce is the online calculation tool, which is one of the most powerful functions on our website. So when we click the CentileBrain model on the top bar, it will show up the generate normative deviation values from your sample. The online calculation tool is very user-friendly, so which means it lets anyone apply our model to your own data set, even if you don't have any coding background. So you don't have to worry about how the script works. And this tool is designed to handle different brain regions and is compatible with both male and female. So to start with, we need to enter our email to finish the registration. So here, for example, I put the test email address here, and I would like to mention that the email address is the only information we collect from the users. So for any other data, we do not collect and we do not access to it. So users don't have to worry about the data privacy issues. So after we put our email here, we can simply follow the 5 simple steps. The first, we need to use FreeSurfer to extract morphometric features such as the subcortical volume, cortical thickness, or surface area based on the on different atlas. And the second, once we extracted these morphometric features, we need to download the provided template here. So when we click this link, it will download the template. And let me just show the template here. So in this template, we can see a lot of columns, and we ask a user to move the extracted FreeSurfer outputs into this template, and we ask users to do not change anything on the column names and column orders. So just keep the template as it is. After we move our data into this template, we can move forward to the next step to upload our Excel file. So here I prepared for a sample test set. Let me just upload it here. I use the subcortical volume female test set. So when we select, it will upload to this calculation tool, and we can preview the uploaded dataset on the right panel here for accuracy check. And we can see this dataset includes data from different sites, from UBC and Mount Sinai, et cetera, but all these data are fake and just for test purposes. And after we make sure that this is the dataset that we want to use for calculation, we can click compute button here to start the calculation. So I would like to mention that the compute button will not active until we can preview our dataset. So which means we need to wait to see the uploaded dataset on the right panel to start the calculation process. And the calculation process normally takes a few seconds or a few minutes to finish. It depends on the data size and the choice, the model we choose. Like typically for subcortical volume, it is the fastest one, and it can take up to a few seconds to finish the process. While for the cortical thickness and the surface area, it may take longer time, up to a few minutes to finish everything. And when the calculation is finished, we can see the message up here. The computation is done. Please download the results. And when we comes to the last step, download the result. And as we can see the download file here. And just to quickly show what the result look like, I would like to share this slides here. So this is the result that we just we got from the online calculation tool, and it includes 7 different files, including the predicted scores, predicted scores, centile scores, Z scores for all breed regions for each individuals, as well as 4 key metrics we used to evaluate your result, which includes the correlation, explained variance, mean absolute error, as well as the root mean squared error. So let's take a look at each of them and what they look like. So the first is the prediction, the predicted score. The predicted score is just the predicted value from using our CentileBrain model. and it includes the predicted score for each brain region and for each individual. The order of rows remain the same order that from your uploaded file, so you don't have to worry about the order. And the next is the Z-score. The order remain the same as the order you uploaded, and the Z-score, like the predicted score, it is also for each individual and each brain region. The next is the centile score. You might notice it's slightly different because from our uploaded data set, it includes many more subjects. But for this, we see less individuals here. This is because the centile scores will only generate for the unique age in your uploaded file. So for example, if the uploaded file has 100 subjects, but only 30 of them are unique ages, then the centile values will be generated only based on those 30 unique values. And in the output, in the centile value output file, they will be sorted by ascending order, like we see here in the first column. And the next 4 slides is just the 4 metrics that we use to evaluate our results, including the correlation, explain the variance, the mean absolute error, and the root mean squared error. So just a quick look at the result of this online calculation tool. And back to our website, we strongly recommend and we ask everyone to use this online calculation tool to download the instruction here. This instruction includes the full details on how to use this online calculation tool. So for some people, they wonder whether we can use the script instead of this online calculation tool to calculate some scores. Yes, we do. We do provide our script. So when we switch to the CentileBrain on the left, this is the script that we provided for users, and users can use this script to generate their result based on our pre-trained CentileBrain model. And some people may also wonder whether they can train their own models by using their own data. The answer is still yes. So when we click the tutorial on the top bar here, we provide different versions for different morphometric features. So each of these script allows people to generate their own CentileBrain model by using their own data. So for these scripts, you will get your own trained model and use your own trained model to generate your own results. So for more details, you can refer to this page, and yeah, it's pretty specific. So this is everything that I have for today for some of the important features on our CentileBrain website. And thank you everyone's attention, and any questions are welcome during the Q&A session. Thank you very much.